So today we are going to discuss about amino acid catabolism. Catabolism of the amino acids are specifically done to remove amino group from the amino acid, which is ultimately disposed of in urea synthesis and excreted from the body, as urea itself is very toxic for the body. Carbon skeletons, which are produced from the standard amino acids after removal of amino group from amino acid, is entering into tricarboxylic acid cycle, as it is intermediates or their precursors, and it is metabolized finally to carbon dioxide and water, or used in the gluconeogenesis. Catabolic pathway of amino acids involves three common stages, namely removal of alpha amino group from amino acids known as amino acid deamination. In amino acid deamination, there is conversion of amino group into ammonia. Second stage is incorporation of ammonia into urea. And third stage is conversion of amino acids carbon skeleton, which is common metabolic intermediate, into either TCA or gluconeogenesis. Now you can see this diagram showing general pathway of amino acid catabolism. Here it has been mentioned that protein is broken down into various free amino acids by action of proteolytic enzymes. And this free amino acid, after deamination, is converted into alpha keto acids and amino group. This amino group coming from various amino acids is utilized either for biosynthesis of proteinaceous factors to be used in the body, or it is excreted in the form of urea from the body. Second part of amino acid, that is alpha keto acid, enters into citric acid cycle for production of ATP and ultimately water and carbon dioxide. Amino acid deamination. The removal of the alpha amino group from amino acid involves two types of biochemical reactions, that is transamination and oxidative deamination. First of all, we will discuss about transamination. The dominant reactions is involved in removing amino acid nitrogen and is known as transamination. This class of reactions funnel nitrogen from all free amino acids into a small number of compounds, which is either oxidatively deaminated, producing ammonia, or their amino groups are converted to urea by the urea cycle taking place in the liver. Transaminations involve moving an amino group from a donor alpha amino acid to the keto carbon of an acceptor alpha keto acid. Alpha keto derivatives of amino acid and corresponding amino acid forms. All amino acids except lysine, threonine, and proline. Mind it, these three amino acids are exception of this process. Lysine, threonine, and proline. Except these three amino acids, all other amino acids participate in transamination during catabolism. Transamination is readily reversible reaction. This reaction is catalyzed by the enzyme called aminotransferase, which is also known as transaminase. Each aminotransferase enzyme is specific for, as usual, each enzyme is specific for its substrate. Similarly, aminotransferase is also specific for one or at most a few amino group donors. Aminotransferases are named after the specific amino group donor because the acceptor of the amino group is almost always alpha ketoglutarate that gets aminated to glutamate. Here in this reaction, you can see in transamination, there is a reaction between amino acid and alpha keto acids. That amino group of amino acid 1 is being transferred to alpha keto acids, and this amino acid is being converted into alpha keto acids, whereas alpha keto acid taking that amino group is converted into amino acid 2. This is process of transamination, transfer of amino group from amino acid 1 to alpha keto acid 2. Aminotransferases require participation of an aldehyde containing coenzyme pyridoxal 5-phosphate, a derivative of pyridoxine, that is vitamin B6. Pyridoxal 5-phosphate is covalently attached to the enzyme via a Schiff-base linkage formed by the condensation of its aldehyde group with the alpha amino group of lysine residue. Aminotransferases act by transferring the amino group of an amino acid to pyridoxal part of the coenzyme to generate pyridoxamine phosphate. The pyridoxamine form of the coenzyme then reacts with an alpha keto acid to form an amino acid and regenerates the original aldehyde form of the coenzyme. The most common compounds involved as a donor or acceptor pair in transamination reactions are glutamate and alpha ketoglutarate, which participate in reactions with many different amino transferases. Here you can see amino acids react with alpha ketoglutarate and transferring its amino group to alpha ketoglutarate. Amino acid is converted into alpha keto acid and alpha Kg is converted into glutamate. All the amino nitrogen from amino acid that undergoes transamination can be concentrated in glutamate because L-glutamate is the only amino acid that undergoes oxidative deamination at an appreciable rate. Here there are so many examples of transamination, and in each reaction you will find formation of glutamate. In first reaction, there is reaction between alanine and alpha ketoglutarate, and in the presence of alanine transaminase, alanine is being converted into pyruvate and alpha Kg into glutamate. In second reaction, aspartate reacts with alpha Kg in the presence of aspartate transaminase, and aspartate is being converted into oxaloacetate, and finally, just like previous reaction, alpha Kg is converted into glutamate. In next reaction, tyrosine reacts with alpha Kg in the presence of tyrosine transaminase, and tyrosine is converted into para-hydroxyphenyl pyruvate, and in this reaction also, just like previous reactions, finally glutamate is prepared. Now we will now we will discuss about glucose-alanine cycle. In skeletal muscle, excess amino groups are generally transferred to pyruvate, which is converted into alanine and enters into the liver and undergoes transamination to be converted into pyruvate, which enters gluconeogenesis and converted into glucose. Then this glucose returns back to the muscles where it is glycolytically degraded to pyruvate. This we will see in the glucose-alanine cycle. Here you can see glucose enters muscle, and as usual, it undergoes glycolysis and is converted into 2 molecules of pyruvate, producing 2 molecules of ATP. Now this pyruvate in the presence of alpha amino acid is converted into alanine, producing alpha keto acid, which will enter TCA cycle. And this alanine through blood enters liver. Here this alanine is converted into pyruvate, producing amino group, which enters urea cycle in the liver and produces urea, which is being excreted by the body because it is very toxic for living cells. Now this pyruvate, again by gluconeogenesis, is converted into glucose, consuming 6 molecules of ATP, and this glucose again goes back to muscle to repeat this glucose alanine cycle. This is very important for excretion of urea from the body. Rather, this is very important for formation of urea and excreted from the body. Now comes second important stage, that is oxidative deamination. Transamination does not result in any net deamination. During oxidative deamination, an amino acid is converted into keto acid by the removal of the amine functional group as ammonia, and the amine functional group is replaced by ketone group. Ammonia eventually goes into the urea cycle. As the recipient of amino groups from many sources, glutamate now sheds it as ammonia for excretion, and the product alpha ketoglutarate can recycle as a nitrogen acceptor, enter the tricarboxylic acid cycle, or serve as a precursor in gluconeogenesis. Deamination occurs mainly through the oxidative deamination of glutamate by glutamate dehydrogenase, which is allosterically inhibited by GTP and NADH and activated by ADP and NAD+. The reaction requires an oxidizing agent like NAD+ or NADP+. In this reaction, you can see there is oxidative deamination of glutamate. Glutamate in the presence of NADP+ is converted into alpha ketoglutarate and amino group. Here, glutamate in the presence of glutamate dehydrogenase enzyme and ADP and NADPH. In the presence of ADP and NADP+, which is ultimately converted into NADPH plus H+, is converted into alpha ketoglutarate, and amino group is separated from glutamate. Now we will discuss about urea cycle. Living organisms excrete the excess nitrogen resulting from urea cycle or amino acid breakdown in one of the three ways. Many aquatic animals simply excrete ammonia where water is less plentiful. However, processes have evolved that convert ammonia to less toxic waste products that therefore require less water for excretion. One such product is urea and other is uric acid. Accordingly, living organisms are classified as being either ammonotelic, that is ammonia excreting, specifically aquatic animals, fish, etc. Ureotelic, that is urea excreting, specifically mammals including human being, or uricotelic, that is uric acid excreting, specifically lizard, insects, birds, etc. Urea is formed from ammonia, carbon dioxide, and aspartate in a cyclic pathway referred to as the urea cycle. Because the urea cycle was discovered by Krebs and Henseleit, it is often referred to as Krebs-Henseleit cycle. Urea synthesis takes place exclusively in the hepatocytes, that is liver cells, which consists of 5 sequential enzymatic reactions. You should remember that first 2 reactions exclusively take part in the liver mitochondria, and remaining 3 reactions take place in the cytosol of the cell. Now in urea cycle, there is formation of carbamoyl phosphate in mitochondria. The substrates for this reaction catalyzed by carbamoyl phosphate synthetase 1 are amino group and bicarbonate group. Because two molecules of ATP are required in carbamoyl phosphate synthesis, this reaction is essentially irreversible. Now carbamoyl phosphate reacts with ornithine to produce citrulline, which passes into the cytosol. Next, three steps that occur in cytosol involves formation of argininosuccinate by ATP-dependent reaction of citrulline with aspartate. Formation of arginine from argininosuccinate. This reaction releases fumarate, which enters citric acid cycle, and formation of urea and regeneration of ornithine. Now here we can discuss about urea cycle. First reaction is formation of carbamoyl phosphate from ammonia, carbon dioxide, and in the presence of ATP. Here ammonia combines with carbon dioxide derived from decarboxylation reaction to form carbamoyl phosphate in the presence of biotin and acetyl glutamate and two molecules of ATP. Biotin activates carbon dioxide and an acetyl glutamate acts as a cofactor for the enzyme carbamoyl phosphate synthetase 1. Then this next reaction is formation of citrulline. Citrulline is formed by the transfer of carbamoyl group, that is CONH2, from carbamoyl phosphate to ornithine. Carbamoyl phosphate to ornithine. This reaction is catalyzed by the enzyme ornithine transcarbamylase enzyme. Then next step is formation of argininosuccinic acid. Here, second molecule of nitrogen required for the synthesis of urea is provided by the amino group of aspartate or aspartic acid, which condenses with citrulline to form argininosuccinate. This reaction is catalyzed by argininosuccinate synthetase and requires a molecule of ATP. Then there is formation of arginine from this argininosuccinic acid. Reversible cleavage of argininosuccinate takes place to form arginine and fumarate. This fumarate enters tricarboxylic acid cycle, and this reaction is catalyzed by argininosuccinase. Then there is a formation of urea from arginine. It is the last reaction of urea cycle which generates ornithine from arginine from its re-entry into the cycle. Urea is synthesized in the process from the hydrolytic cleavage of guanidino group of arginine by arginase. And from arginine, as urea is separated, remaining compound is ornithine, which again takes part in next cycle of this urea cycle. It continues as such for next cycle. And for next cycle, it is clear from the above mentioned scheme of reactions that for the synthesis of one molecule of urea, a mole each of ammonia, carbon dioxide, and alpha amino nitrogen of aspartate and 4 moles of ATP are consumed. Net reaction of urea cycle involves carbon dioxide plus amino group plus aspartate plus 3 ATP and 2 molecule of water is converted into formation of urea, fumarate to ADP and AMP. That's all about protein catabolism.